The overall goal of this procedure is to image cell behavior within the embryonic neural tube at high resolution for long periods of time. This is accomplished by first transfecting the early neural tube with a construct driving expression of a fluorescent protein of choice to mark single cells. The next step is to make slices of the early embryo and mount them onto glass-bottomed dishes. After recovery in an incubator, the embryo slices are imaged on a wide-field microscope at regular intervals. Ultimately, this technique can be used to study cell behavior within the developing neuroepithelium over long periods of time, with high spatial and temporal resolution.
The main advantage of this technique over existing methods for imaging living tissue is that it allows us to observe individual cell behavior over long periods of time at high spatial and temporal resolution.
This method allows you to address key questions in the field of developmental neurobiology, such as the role of mitotic spindle orientation in cell fate choice, or how signaling dynamics might be regulating cell behavior.
Prior to electroporation of plasmids into the neural tube, glass needles were prepared using a microcapillary puller. Under a dissecting microscope, use fine forceps to break off the tip of the needle. The end of the needle should be sharp enough to pierce the neural tube while not being so narrow that it impedes injection of the DNA solution.